Welcome to SeedWorld's Strategy webinar. Today we're going to try and answer the question, can seed treatments improve water use efficiency? My name is Julie Deering. Many of you know me as the editor of SeedWorld, and today I'm happy to be your host. Before we begin, I'd like to take a moment to thank our generous sponsors, BASF, Bayer, and Syngenta for their support. I also want to let you know that this webinar is being recorded and will be made available on seedworld.com following the event. As we go along, you'll likely have some questions for our speakers. I just ask that you simply type these into the chat box and we'll do our best to address them during the Q&A session. Today's speakers are Bruce Battles of Syngenta Seed Care and Jesse Deerdorf of Indigo. During our time here, they will discuss the role of seed treatments as it relates to plant health and what this means for water access. A seed treatment that promotes vigor, helping plants better cope with adverse growing conditions such as drought and heat stress. And finally, a new microbial seed treatment that's making waves for cotton growers and what it could mean for other crops. After the presentations, we'll open it up to questions. So let's go ahead and get started. Our first speaker is Bruce Battles, who serves as the Seed Care Technology Manager for North America, where he is responsible for the development of new seed-applied technologies that protect crops from disease, insect, and nematode damage. Prior to his current role, Bruce served as the Solutions Development Manager for corn. Bruce has worked with Syngenta for 15 years and held a number of roles, including research agronomist, product development agronomist, technical sales agronomist, and agronomy marketing manager. Prior to joining Syngenta, he spent time as a research assistant at Iowa State University, working in the area of weed science as well as retail agronomy. Bruce earned his master's in crop production and physiology from Iowa State University and earned his bachelor's degree in agronomy from Oklahoma State University. He was born and raised in Oklahoma and has lived and worked in Iowa since 1996. Bruce, thanks for being here with us today. It's all yours.
Thank you for the introduction. It's good to be here today, and look forward to having questions and addressed at the end of this. So we'll start with today as the topic that was laid out and we were asked to talk about is, can seed treatments help plants use water more efficiently? So we really want to take a step back and take a look at the bigger picture of what the challenge is, right? And so from water is obviously our biggest limiting factor in agriculture, and likewise it gives us the ability to feed the world and a growing population. So with this, we really have to concentrate on how we're using water and be more efficient about it so that we can grow more and be safer and better quality of food as well to meet all the future demands. So with that, we have a big challenge ahead of us, but we really see a lot of opportunities, a lot of new technologies that are constantly being developed that we feel that we can comfortably meet this challenge and improve yields while also enhancing the quality and safety of food supply through more efficient water use. So with that, though, we really need to take a step back and think about how the plant uses water prior to really diving into exploring water use efficiency and different seed treatments that can help with that. And there are a lot of ways the plant uses water, from driven from the climate or the soil or the crop development stage. And when we think about climate, we often think about the amount of rainfall that falls in different areas and seasonal throughout a year, and associate that with drought stress. But there's really, even within those areas, there's differing amounts of water availability driven by temperature, humidity, solar radiation, and wind speed, because a lot of water is actually lost through evapotranspiration out of the plant, because the plant is using it as not only a method for increasing photosynthesis, but also using it as a method to cool the plant under those extreme temperatures. So there's a lot of water that's lost directly into the atmosphere. And that's going to go up with temperature, and as humidity levels go down, the plant needs more and more moisture to cool itself as it goes through as well. And as wind speeds increase, that's also going to increase that demand amount of water that's lost into the atmosphere. Secondly, along that, it really depends on the crop stage of development as well. That really influences the amount of leaf area that's visible in the plant and exposed, which is a good thing to drive photosynthesis and build the factory that the plant's trying to develop in order to produce higher yields. but along with that, as the more leaf area you have, the more water losses you're going to have, more use and consumption. Rooting depth also plays a big factor into that, and even things like crop relative maturity can impact the timing of the year of peak demands and things that we consider. And lastly is soil, really. So soil affects in several ways. So from a fertility level, the higher fertility level you are, you're going to get more rapid growth and more development above ground, which is going to lead to higher demands of water. Along with that, impacting the soil impacts the water holding capacity, so the amount of water that can be readily available to the plant at any given time is really dictated by the soil type that goes in. And as well, the amount of water intake rate. So the soil itself cannot buffer that or hold moisture and hold it available to the plant if it can't get it into the soil. And different crop rotation practices, as well as previous crops, as well as the soil type itself, will all impact the amount of water and the rate that soil is able to absorb water and take it in. And it's really not one in particular thing that we talk about on the slide, but it's really the interaction of all three of those various areas that makes it a really complex and difficult issue to manage as we go through and thinking about water use efficiency. As most people are familiar with the graph on the right, we know and commonly associate the peak water demands as we go into the flowering crop timing frame for the crop. No matter what crop it is, this is an example for corn that we're looking at here, but really the peak demand is going to occur right going into the pollination and start to begin to decrease as we go into kernel fill and stages as well. But the most critical and the most negative impact we can see on the crop from a yield perspective is water losses or water availability shortages throughout those reproductive time frames of the plant. What we often think about when we're thinking about water and crop use, we really like to concentrate on that area for those logical reasons, but we often overlook what value of the early stages or what we do to set that plant up for success later during those vegetative stages can help influence the water availability later on in the plant. And so that's really what we want to talk about in the next few slides, is talking about new technologies and examples that we can put forward to help operate more efficiently. For example, early in the season, we're all familiar with soil-borne diseases, but we don't often think about them in the context of making a more efficient plant. So often we think about things like Pythium, as well as Phytophthora in soybeans, that are really diseases that limit the plant stands. And we can commonly associate that with yield by having stand reductions in yield, but we often overlook the nibblers, you might say, of the pathology world. So fusariums, the rhizoctonias, that don't always decrease plant stands that we can see as we're walking fields, but do commonly decrease the efficiency of the plant. So they're known for reducing those root hairs that are taking up nutrients and moisture in the soil. And any of those types of reductions lead directly to later in the season how efficient that plant can be. Another one that's commonly overlooked is nematodes. It's not a pest that we can dig up and see readily, right? And we can do soil tests, we can understand if they're there or if they're not there, but it's really hard to associate the value with that and how that relates to an efficient or inefficient plant. And it's really because we've had very few tools to gauge how valuable controlling that is up until recently. And so, but all in all, whether it's a disease like Fusarium rhizoctonia or a pest like nematodes, it's really extremely important to have a healthy plant and set that plant up for that stage early in the season. And those are all things that we probably have thought about at some point in time, but we don't always think about the lower half portion of the slide. So what's in their relationship into water use efficiency. So when we think about how quick a plant will develop a canopy, how does that relate to water use efficiency? So a couple of things that really drives, right? The slower that plant is developing, that's reducing our ability to have good weed control in some situations. And along with that, we get increased competition between the plant and those weeds in the field. And that competition can be for nutrients and light, but it can also be for water as well. Along with this, that earlier canopy really, by closing the canopy earlier in the season and quicker in the season, we can have a lot of water that is lost from the soil directly, so evapotranspiration that's occurring in the soil. And so by canopying the plant earlier and covering the soil over and shading that, we can reduce the amount of evaporation coming directly off the soil, which is a lot of times 20% of the total soil moisture losses or use in general is coming from just evaporation. So, and then finally, the early season canopy is really driving the leaf area that's setting yourself up for the photosynthesis that's coming later in the season. But really all the things on the slide that we talked about, from the traditional thoughts around managing diseases and pests to getting a quicker developing plant that canopies earlier, are all important, and we've got a lot of background history on how seed treatments themselves can help mitigate some of these different stresses. And so that's really what I want to spend the remainder few slides and the time frame we have here to address exactly the science behind what we're seeing with seed treatments and how they're impacting the plant and affecting those areas. And the first one I really want to start with is a product branded as Vibrance. The active ingredient in Vibrance is Sedaxane. Sedaxane, or Vibrance, is a fungicide that is commonly used across multiple crops, and it's labeled as a fungicide that's managing diseases because it's a SDHI inhibitor, which is a fairly new mode of action in the chemistry world. And one of the unique things that we've learned as we've brought this molecule to work is that not only does it have specific activity on diseases, which it's really focused on rhizoctonia. Example, it's a premier product for controlling and managing rhizoctonia, but it also indirectly has other benefits. And this is an example of a slide that I'm showing from the European Root Health Forum that occurred in 2015. And it was put together by Dr. Ray in the University of Nottingham. And there's actually a paper you can see in the lower right-hand corner to refer back to specifically on this and read more about it, and how they've discovered that Sedaxane, or Vibrance, alters the gene expression, which in turn is increasing the photosynthesis efficiency of the plant and growth, and as well as improving the tolerance to drought. And in this example, they used wheat as their crop that they explored more. And so some of the key findings that they have found in this paper is really that, one, we already knew how important photosynthesis is to the plant, and we understand that under drought stress, photosynthesis is one of the first things that are compromised, leading to those yield reductions that we talked about. And so anything that can help preserve photosynthesis through times of drought could potentially help them and help the plant operate more efficiently from a water use efficiency standpoint. They also understood from their findings in this paper that Vibrance, when applied as a seed treatment on wheat in this example, increased that photosynthetic efficiency. So as a result, that's improving the biomass production as compared to an untreated check when under drought type of conditions. So the study really indicates that the Vibrance can promote that continued growth and development under drought stress. And the way it does that really is by redirecting metabolites from a defensive stress response type of action towards a growth and adaptive development action. So essentially, in layman's terms, what it's telling the plant is that, hey, plant, you can be a little more conservative. You don't have to be as conservative under this period of drought stress. Continue your growth and development more normally like you would, and don't shut down quite so quick. And that's the layman term for what this molecule is doing besides just the fungicidal properties. Another example that we've seen, a little bit older example, but we've seen a lot of field experience with it, is understanding how we can also see physiological changes from products like Cruiser. So Cruiser is a neonic insecticide, a thiamethoxam, that's been labeled for several years, and we continually are learning more and more about how that is impacting the plant. We immediately knew the insecticidal components of it, but we are also understanding more and more about what it's doing to physiologically change the plant and help with some of the things we talked about earlier, like driving to quicker, earlier canopy. And these are just some examples here showing you how a root on the left is impacted through that. And then the main trigger mechanisms that we've seen is an increase in cell division, so increase in the numbers of cells that are in the root that are being developed, as well as increased elongation of cells, so increased size of the cells, that's helping get to a point where we can get a quicker canopy to help with weed control or conserve moisture, and even just in some cases that are hard, difficult to establish type of situations with crusting and things like that, to break the crop through that and emerge quicker and faster and get more established quickly. All this leading to a potential for increases in yield. This next slide here really kind of just focuses in on that. So for one, increasing more specifically the size of the cells that we're talking about. And so you can see an example in the top upper picture versus the lower half of the picture. There's a light yellow bar that was inserted into the microscopic image there to measure the length of those cells and comparing the difference between a control versus a Cruiser treated seed and the cell on that, and illustrating the differences. And the next slide here also does a similar thing, but it's focusing on the number of cells that are reproduced. And so in this situation, in the apical meristem, or the root tip, right, we're seeing a higher number of cells that are developing. In this example, when they labeled the tiny green dots, you can see they labeled each of the apical meristem cells. They saw on average an increase of 11 cells per apical meristem as compared to the control without using a Cruiser. So those are a couple of the scientific aspect around proving and validating how and why it's working. But what a lot of our customers over the years have seen is something similar to this, that a quite consistent response in the field showing that early canopy and development. So the left-hand side of this image illustrates the same soybean variety as the right-hand side planted the same day. The only difference is the seed treatment containing Cruiser on the left and versus an untreated on the right. And I think this picture really does a nice job of illustrating how that can relate to weed control as well as moisture conservation. And by looking down the rows on the right, you can see the soil real easily, and it's uncanopied, whereas we've already achieved a full canopy almost on the left-hand side. And in those situations. So a really nice example of that in soybeans, but we've seen the same type of response very consistently across other crops as well. And this is just an example of the same type of response conducted in the controlled environment lab and within sterile soil type of conditions. And you can see the Cruiser treated corn seeds on the right versus the untreated seeds on the left. And a very noticeable difference, hard to measure out, and if you're not there in the field on the right day and have the head-to-head comparison, you may not see it, but it is validated, the increase in growth and development. And a lot of times this is looking at what's happening above ground, but it's also happening what's developing below ground, the plant. And one example of that, because what happens below ground is even more important for how effective that plant is at uptaking moisture from the soil. So this next slide really looks at kind of an extreme situation where we put trials out under drought type of conditions, actually in Yuma, Arizona. And we looked at a couple of rates of Cruiser as compared to a non-Cruiser treated corn seed in that situation. What we're looking at is the fresh root weight. So 30 days after planting, we went in and dug up 20-something plants per treatment, took them back, cleaned them off, weighed the plants, and evaluated them. And this is showing what happened in those situations. Actually, across 3 different irrigation regimes. So the one on the left is a 50% ET irrigation scheduling, evapotranspiration. The middle 3 bars is what happened at 75%, a little bit higher moisture irrigation scheduling, and the upper right is meeting that full plant's demand by irrigating to 100% evapotranspiration. And the nice thing is you see the similar trend across all 3 of those different irrigation regimes. You can see a really nice rate response as we continue to increase that rate of cruiser. We also continue to get an increase in root weight and fresh weight, which would lend us to believe that that plant's more efficient at being able to tap out and uptake any available moisture in the soil. So the last slide I really want to show here is also, we talked a lot about soil moisture and uptake and water use efficiency, but the nice thing about cruiser is what we've also documented is that it helps the plant through other stresses as well, besides just a drought stress. And we've identified this back to a specific detoxifying enzyme that's naturally produced in the plant. And what we've learned is GST, or glutathione S transferase, is that detoxifying enzyme we're discussing here. And if you look at the bar graph on the right, what that's showing is that as you incrementally go from left to right on the graph, those are increasing rates of cruiser that's been applied to the seed. And what we're seeing is that as you increase that rate, we're getting higher levels of GST, or glutathione S transferase, within the plant, and which is commonly linked with helping detoxify that plant from stresses such as cold temperatures, salt stresses, or even acidic soil type of conditions. So we know that anytime we have cruiser applied, we can help overcome those types of levels of stress. So just to wrap up today, we went through a lot of different information. What does it all really mean at the end of the day? I think that some of the key points is that from a seed-applied fungicide standpoint, we can protect those young plants from these nibblers, Rhizoctonia, Fusarium, soil-borne diseases. And indirectly by doing that, or directly by doing that, we can make that plant more efficient for later in the growing season when the peak water demand occurs. Likewise, in there, by using seed-applied nematicides, we can also help make that plant more efficient later in the season and better able to fully extract the demand that it needs for soil moisture and nutrients. But in addition to those more traditional approaches and thoughts around seed treatments and what their normal roles would be, we'll also discussed a lot of things that are in addition to that pest protection. So some of the things we talked about was how some active ingredients like vibrants as well as cruiser can help germinate and establish the plant under stressful conditions. They can help make the plant more robust and develop more efficient roots that can help later in the season. We can get an earlier canopy, which can indirectly benefit soil moisture use as well. And then with the vibrance example, we learned that we can help maintain growth at a more normal rate during drought conditions versus letting the plant start to shut down and stall out. So what that really means to our customers, our growers that are using the products in the field, that means a more healthy, more efficient plant that can hopefully better manage stresses later. And with that, it provides more yield stability out in the field. And so we haven't shown any yield data in this presentation, but that's really what our yield data has shown, especially with products like Vibrance, year in, year out. We see continual bumps in yield, even in low disease pressure type environments that go back to the modes of action, the scientific information that we talked about before. And then finally, what I would like to close with is seed treatments, again, I would like to stress that they're just another tool in helping mitigate water stress. And drought is very complicated. The timing that it occurs, the genetics interact with drought stress, other tools of management traits, the GM traits, as well as newly being developed traits that come to market, and even the herbicide and fungicide programs can help with that to mitigate that stress. And so seed treatments is just another tool that's required to help manage that, but it does require the full toolbox in different areas to help alleviate or offset those risks. So with that, that's all I really wanted to cover today, and we'll hand it over to my colleague to continue.
Thanks, Bruce. So what I am hearing you say kind of in closing thoughts is that we can use seed treatments to physiologically change the plants so that they can better handle water stress and have more access to the water that is in the soil profile.
Exactly.
Okay, well, thank you so much. Our next speaker is Jesse Deerdorf. Jesse grew up near his family farm, Deerdorf Sweet Corn, Inc., in Adel, Iowa. The farm is still family-owned and currently supplies the Des Moines area with quality sweet corn. Jesse's career started with an internship at Bayer, where he was involved in the development of Poncho Votivo, and ever since he's been working on specialized seed treatments with a focus on biologicals. As a field development specialist at Becker Underwood, his efforts were concentrated on the Advanced BioStacked program. After the Becker Underwood sale to BASF, Jesse led several seed treatment studies and is now the product management lead at Indigo, a company dedicated to using beneficial microbes residing within plant tissues that optimize crop health and improve productivity. Today I've asked Jesse to share with you a new seed treatment for cotton and how this new knowledge can be used in other crops. Jesse, thanks so much for being here with us today, and I'll let you take the reins.
Great. Happy to be here. Thank you for the introduction. And on behalf of Indigo Agriculture, I'd like to thank Seed World for the opportunity to participate in the webinar today. We know there's been a plateau in crop yields over the past years. The increasing unpredictability of weather is one of the biggest challenges growers are facing today or in the future. Whether it's temperature extremes, too little or too much rainfall, or depletion of land quality, altogether they determine a need for innovation. This table illustrates an increasing gap between crop production and forecasted need. This drives the need to explore, in partnership with growers, new technologies that are more in line with consumer wishes. What is a microbiome? It's a community of microbes that live in a common habitat. Microbes can be bacteria, fungi, viruses, both good and bad. They symbiotically help sustain life in all of its forms. Microbes may become the biological software for the plant. If we take a step back and look at the human microbiome as one of the most significant discoveries in the past 20 years. In the early 2000s, the human microbiome was discovered, and a few years later, a $115 million human microbiome project was launched. All this leading to the discovery and importance of probiotics in humans, and not to mention many other medical breakthroughs. So what is its relationship with agriculture? Plants also have microbiomes inside of their tissue. Building on the progress of the human microbiome, the plant microbiome has the potential to be the next innovation revolution in agriculture. Indigo is uniquely focused on in-plant endophytes. Endophytes are organisms, often fungi and bacteria, that live between the plant cells. Most microbial products in crop production have focused on plant soil microbiomes. Indigo has drilled down to unlock the potential of symbiotic endophytes. This is what makes our products unique. We believe that endophytes can create a bridge to achieve the genetic potential of crops. What exactly are we bridging? We're helping the plant overcome abiotic stresses to reach a higher yield potential. And endophytes have a specific interaction with plant signaling, leading to enhanced root growth and increased water and nutrient uptake. Indigo is currently focusing its efforts on abiotic stresses. Abiotic stresses are environmental stresses that negatively impact growth, development, and yield. Indigo's first product launch was in 2016 with Indigo Cotton. Cotton is a highly water-dependent crop, and it's offered us the opportunity to showcase Indigo's product and technology. Indigo research and development is focused on recreating the plant's natural microbial makeup to unlock its beneficial potential. In our Indigo labs, we identify and sequence thousands of endophytes that live inside the plant itself. The resulting products complement a plant's natural process for improving water use efficiency in various stages of growth development. As noted earlier, harnessing native endophytes and introducing them to modern varieties can have a significant effect on water use efficiency. The photograph to the right demonstrates the improvement in overall resilience in the plants treated with indigo products under a low water regime. As mentioned before, we've launched indigo cotton in the spring of 2016 on approximately 50,000 acres. The majority of the acres were planted in West Texas, with the remaining acres being planted in 4 nearby states. This was all accomplished through the collaboration of over 70 growers. The 2016 preliminary data shows roughly an 11% yield increase in both dryland and irrigated acres, with a 75% win rate overall. We also partnered with Dr. Greg Sword at Texas A&M, and he has a key publication written on this study itself, so if you want some more detailed information, feel free to look that up. This is a photograph of a recent seedling assay that demonstrates almost a 10% increase in root biomass when comparing an untreated check to indigo cotton. Increased root mass and root hairs is essential to increasing the water use efficiency and nutrient uptake. What I've said comes down to this. With the utilization of the plant microbiome, and specifically endophytes, indigo products can improve water use efficiency, which brings us back to our overall mission. Indigo harnesses nature to help farmers sustainably feed the planet. Thank you.
Thanks, Jesse. Were you involved in at all in the initial research with Texas A&M and Greg Sword, or where did you come into the mix there?
That actually came in before my time. I joined the company in October, so I believe that kind of started last spring.
Okay. And I was informed that there was some potential application for crossover to other crops. Can you speak to that a little bit in terms of what we've learned from the current research and how it might be applied to wheat specifically?
Sure. Yeah, we've actually this fall, 2016, we launched our indigo wheat for winter wheat. And our products aim to improve water uptake in this crop as well and better defend themselves against drought. So we have observed that plants grown with indigo develop hardier root structure, which can allow for increased water utilization to withstand stress conditions and boost crop productivity.
That brings us to our question and answer session. So if you do have any questions, please go ahead and type that into the chat box, and I'll make sure that our speakers have a chance to answer those. So I think the first question there is for Bruce. How long does the active molecule persist in the seed or seedling?
Okay. Very good question, and obviously depends somewhat on what molecule we're referring to, right? And we referred to a couple of different things like thiamethoxam within Cruiser, as well as the Sudaxane, or branded as Vibrance. And but all in all, from a seed treatment standpoint, we're typically looking at, when we think about efficacy on an insect or a disease, we're looking at a window of 28 to 35 days type of a window after planting, and at that point in time, generally the crop's pretty established for the target pest that you're talking about. So.
Thank you. And then the next question is also for you, Bruce. What was the substrate that was used? Was it direct quantification through size exclusion chromatography, or spectrophotometric absorbance measurements for GST activity?
Yeah, so really good question that I probably don't have an immediate answer for the group. This was a study that was done I think in Europe with the Harper Adams University. I can definitely get more of those specific details around it, but if the individual wanted to email me directly, I could get that information for them, but I don't have that level of detail right now.
And this question is also for you, Bruce. The company, Syngenta, recently announced that it will begin sales of a seed treatment biostimulant called Apivio. What is the mode of action and the benefits of the product related to water use efficiency?
Excellent question. So Apivio is a new brand umbrella that has been launched at a global level within Syngenta. Here in the United States, we have not actually launched any of the Apivio brands within our seed treatment portfolio. We are currently looking at different molecules that may fit underneath that umbrella and eventually bring them out. But to the specific question around the molecule, since it is a branding umbrella, there are different molecules depending on where that product is launched. For example, there's one that's been launched in South America, in the Brazil market, that I think most people are probably referring to. But we also have another one that's been launched in APAC or China, and they actually have different targets. So it's more of a herbicide safener and some of those things that are occurring with the China situation. Whereas the LATAM or South America market, that's really a product that is looking at enhancing nutrient uptake out of the soil and creating efficiency. And through some of that, I don't have all the exact specifics around it, but some of that's increased growth and development, really similar to what we talked about with the Cruiser and some of the examples we showed with Cruiser. And that's the level of detail I have for sharing that now with those specific countries. But again, within the U.S., we don't have any Apivio products that we've currently launched, but we have a team that is looking at those types of products and continually monitoring them and looking for the development opportunities in that area.
Thanks, Bruce. This next question here is for Jesse. Does the indigo endophyte reside in the root, or does it move into the shoot?
So when the seed germinates, the microbes colonize in the plant and multiply. So we're still evaluating some of the translocation throughout the plant, but I would say yes on moving from the roots into the shoot as well.
Great. And let's see here. Bruce, how do you communicate the drought benefit to growers? Is it increased yield stability, and how much yield increase?
Yeah,
if it is yield?
Yeah, so that is a really challenging thing, especially with all the types of products we have, is putting understanding what the value really was after you put them in the ground. And what we've seen through our tests, specifically on this, I'm going to refer more to the Vibrance, the Sedaxing fungicide. And what we've seen over 3-4 years of field testing programs on a broad scale acre, we're seeing a 1.5 bushel to 2.5 bushel type of yield increase range fairly consistently from trial to trial in what we consider broad acre type of trials, so non-inoculated type of conditions, broad acre. And we know that since that target pest for Vibrance is Rhizoctonia, we realize that that's not something that directly impacts yield on a standpoint. It's hard to measure how much that's coming from directly from disease control and enhancing uptake through that versus the drought response that we talked about specifically from the paper and the wheat example. It's really hard to tease out what percent of the time that 1.5 to 2 bushels is coming from which mode of action on that particular molecule. But all in all, what we're seeing, I think to answer your question, is we talk about yield stability. So as you get differing climate type of environments from year to year, you can feel more secure on the yield that you're going to get from that acre by having this product on there.
Great. And then we've got lots of questions coming in here, so this is good. Jesse, what kind of benefits or yield increases were you seeing with indigo and soybeans and wheat?
So actually the wheat product launch was fall-winter wheat, so we haven't seen harvest from that. But in some of our greenhouse assays, I've seen some early plant vigor. And then we have not launched soybeans yet, so.
Okay. And let's see here. Bruce, this is a follow-up question to the one you just got done answering. What stress level do these products work best, mild or severe?
So I'll refer to the Vibrance fungicide as an example, right? And so the yield data we just talked about is kind of that 1.5 to 2.5 bushel range within corn that's really been under what I call moderate to normal type of environments, broadacre across the Midwest. So ranging from the bulk of the data we're looking at is the Iowa, Illinois type of thing. So we're seeing it fairly consistent no matter where we're at, even on the differing levels of stresses. I think obviously with any of these type of products, as your stress level increases and yield potential goes down, there is a point in diminishing returns where it's going to be difficult to do anything, right? So if a plant goes under so severe water stress that puts it in a 50 or 60 bushel yield range, it's going to be very difficult to move the peg 10 bushels or 15 bushels kind of off of that mark. And so everything I refer to is talking more about moderate to normal types of environments without much stress. From a soybean perspective, especially with I'm looking at emergence and stand and everything with Cruiser's example, that's you get some of those severe type of stressful environments where you have crusting and those type of things. That's really where you're going to see a lot of those abiotic stress type of benefits come through. But we can again see, the benefits from controlling pests as well across broad acres and depending on their presence.
Okay. And these next two questions here are for Jesse. What is the goal of cotton acres that indigo will be planted for in 2017? And the same question for wheat this fall.
Okay, thank you. Really can't share our sales goals or acreage goals for the upcoming crops, but I can say they're going to be significantly larger than what you saw last year.
Okay. And in terms of wheat, are there regions that you're targeting, or just as much as you can?
Yeah, so the regions would be Kansas, Illinois, Indiana, southern parts of those states, and then also the southeast, Virginia, Georgia,
okay.
Those regions.
And are you selling the indigo product directly?
No, it is our strategy to take a direct-to-grower approach. This allows the farmer to be more profitable using a sustainable technology.
And question from the same person, how stable is a microbial product?
Yeah, the shelf life of indigo products coincides with the grower planting timelines. So we're making it stable enough to meet those timelines.
So is it a treatment that is done on farm or planted immediately, or does it have like a 30 or 60 day shelf life?
It kind of depends. It depends on the crop. Pre-farm, I guess, is what I would say, but it is treated close to planting, is I guess the best I could put it.
And then what is the intended channel to market in wheat after initial product launches? Secondly, how will that vary from crop to crop?
Because different crops are treated at different times in relationship to planting, so that's how it varies from crop to crop. And I don't want to get too involved in our direct-to-grower strategy because that is somewhat proprietary.
Okay. And I believe this next question is for Bruce, talking about vigor. How do you define early vigor for wheat, and then germination, emergence, and height?
So yeah, there's a variety of ways we use and within Syngenta to determine vigor. We measure the emergence rate, so time frame from planting till one leaf stage, for example, or one collar. We've also used quite a bit a dry weight, so at, for example, 30 days out after planting, digging plants up, cleaning them up, and either doing a whole plant or just the roots or both, and after drying the plant down, measuring in grams per plant type of basis. But we do, as a regular thing, we'll also just do a more subjective approach to rating vigor in field trials as well, commonly, even if we're not doing any of the others, using a 1 to 9 scale. But most generally, if we're publishing something in a paper or whatever, it's going to be based off the dry weight of the plant and root and biomass.
Thanks, Bruce. And Johnny, I'd also like you to answer the same question. How do you define vigor for wheat?
I think it's going to be pretty similar throughout the industry. So we do rely on height in some circumstances, but mostly dry weight and root biomass as well. So.
And when you talk about indigo's response to abiotic stresses, do you plan to work with abiotic stress in the future, such as insects or nematodes?
Yes, most definitely. We're looking in both abiotic and biotic stresses. As eventually we'll need to cover those to make the grower more profitable and be more productive, I guess I should say.
Gotcha. And I think there was a little bit of confusion there between abiotic and biotic, but I think the point came across okay. For Bruce, what does the Syngenta pipeline of new novel products look like in seed care? Good question.
Yeah, so I appreciate that question. We weren't able to cover any of that in there. So we're actually at a real exciting point in our time within Syngenta. We're right at 37 years of seed care within Syngenta right now, and that's brought several molecules to the marketplace that we know, but it's difficult to keep bringing new products and refreshing that portfolio, but we're at a point now where we've got 4 to 5 new molecules that are at or close to going into a registration phase at different timings. So products that will hit the market between 2019 and 2023 type of a time frame. A few of those that we're working a lot on is one of them that's closer to the marketplace is a second mode of action for oomyces, so your pythiums, your Phytophthora type of water mold type of diseases. We're seeing cases, isolated cases that have come up and where the methanoxam or metalaxyl-based products that have been around year in, year out, especially where they've been used in furrow at higher rates, may be having a few isolated cases of resistance pop up. Not really seeing that on a broad acre in corn, soybean production areas, but more in specialty crop areas where they're rotating with potatoes and things like that. So we're trying to stay ahead of the curve on that, and we've had apron as our oomyces on the market for most of that 27 years, and it's still holding up really, really well. But we know at some point in time it's going to need additional help, and so we've got a really broad spectrum product, oomyces, because when we talk about Pythium in general, we're not just talking about one species of Pythium. There are multiple. You may run isolates and get 20 or 30 different species out of a given field. And so having complete broad spectrum Pythium control is really important, and we're seeing that with that molecule. That's going to be a 2019 type of time frame. We've also got a molecule coming to the market for the soybean market that will be very focused on sudden death syndrome, which has been a lot of topic in the industry over the last couple of years. I would say this is more of a step change type of product, and I would put it against anything competitively that is on the marketplace right now as well, from an efficacy standpoint, but also we're seeing really, really good crop safety. So we're not compromising emergence or any visual symptomology of the plant to get that later SBS control with that. So a real safe product. And that, again, is a 2019 type of time frame. And then a little bit farther out, we have a product that is what we consider a next generation nematicide. And it's really a step change over our current. We have two products, one for the soybean market, a Chloriva PN, as well as more of a broader spectrum nematicide for use in the south outside the soybean cyst nematode market, called Victa. This is really a step change type of product against those broader spectrum and higher level of efficacies. And so it'll be looking at a broad registration across almost all crops, corn, soy especially, as well as cotton in the south. And that is going to be a 2023 type of time frame for the most part when we talk about that. So that's really kind of what our some of the key products that are coming in the near future. I guess the one other one we have is to address the wheat market. We have a new insecticide that looks really phenomenal from a wireworm perspective. So the people that may be from the Pacific Northwest have a really good appreciation for how severe of an issue wireworm has become in some of those acres. And so we've got a molecule that has an extremely high efficacy on it, whereas most of the neonics that are used right now don't actually control, just more of act more as a repellent for the wireworm. So populations can continue to build up over year in, year out, and still see damage build up. So we're really excited about that insecticide. It'll be coming again also in that 2023 type of time frame as well to address those needs in that marketplace.
Okay, thanks, Bruce. I've got time for one last question here, and the one that came in is that in addition to seed treatment, is Syngenta coming up with a foliar product molecule for drought mitigation?
So yes, Syngenta is looking at multifaceted approaches. So we have the seed care products we talked about. We also have a native trait approach within corn that we marketed under the brand Artesian that we've been able to measure really consistently under drought stress type of environments and different varying irrigation regimes, the benefits of that native trait as well. And in the background within our trait development group, we also have a continual program looking at GM-based type of traits as well to bring in there. And it's really critical, we feel like, to have a multi-pronged approach for this because it is such a complex beast when we talk about drought. And when you have drought alone, it's a multi-gene type of impact. So just to come at it with a GM trait at one specific gene that you're targeting, it becomes very difficult to have a comprehensive solution. So that's why starting with a seed treatment, maybe a foliar type of approach eventually, which I failed to mention, but we do have a foliar approach we're looking at that's deeper in our portfolio timeline-wise. I don't have those exact timelines today, but there's some collaboration work at a university in California. So.
Great. These have been great questions that have been asked, and hopefully you've been satisfied with the answers that both Bruce and Jesse have provided. I do want to quickly thank Bruce and Jesse for taking the time to join us and sharing their expertise with us today. And I, as always, want to thank our attendees for participating, and I hope you found this information of value. I'd like to quickly, again, thank our sponsors, BASF, Bayer, and Syngenta for their support. And I'd also like to recognize my team behind the scenes, Kyle Dratawani and Teresa Kershowitz, for all their hard work in making this a successful event. Again, this presentation will be made available online at seedworld.com. That's it for today. We hope you have a wonderful rest of the week. And this is Julie Deering of Seed World, signing off.